Good afternoon, everyone, and thank you for attending this new webinar from the Net Zero Center at King's College London. My name is Grazia Toreschini, and I'm coordinating the series. By now, many of you probably know that we have webinars every 1 or 2 months, and we try to have speakers from King's, but also from outside King's to try to develop interdisciplinary research around net zero. So today we are co-hosting this seminar with the King's Climate and Sustainability group to explore King's ambition and integrate globally impactful climate research and action. So today we have 3 speakers in the webinars. So initially, Franz Berghold will introduce KCS and the funding opportunities available, and then we will have 2 speakers from KCS seed funding. So I think without further ado, I will give now the stage to Franz to introduce KCS.
Thank you very much, Grazia. And if I could share my slides, I guess that means you're going to unshare, is that right? Do that. Are you able to see that? Yeah. Okay. So this is a very, very quick overview of what we are. King's Climate and Sustainability, created, funded at the end of 2022, really started operations in the summer of 2023. So we've been going about 2 years now, and I'll try and quickly explain what it is. It was a 3-year transformation program that is due to finish next summer 2026, although we are now talking about an extension, which we think is justified. And the aim was really to drive forward the pace of research, education, and action at King's in relation to environmental sustainability, social sustainability as well, but really trying to move up a level of our activities and our impact. It received nearly £6 million worth of funding from the central SCIF investment fund of the university. So it's a serious investment that the university made here, believing that it was a really important aspect of a higher education's activities and responsibility, really, in a turbulent world, to equip our students, our graduates, with awareness and skills and confidence to deal with these big challenges, global challenges, to grow our research and also to walk the talk, so to make our own operations more sustainable. So the strapline for the whole thing, ambitious, but that's what you need, is to embed sustainability into everything we do. So starting with the academic mission, which is obviously to do with education and research, but also into the way in which we deliver all of those benefits in society. So we organize the work of KCS broadly in four kind of buckets. One is to do with research and education. So we have, the way we're organized is that we're a small team, organizationally situated within the Vice Chancellor's office. I'm called the Assistant Principal for Climate and Sustainability, and my manager in this job is Rachel Mills, who's the Senior Vice President Academic. So that's the way it works. It's quite a sort of the top level of the university, really, and it's seeking to implement a strategy which is coming from the centre. And then, of course, we do quite a lot of communications, engagement and events as part of KCS, and we are engaged very much with the with the directorates, with the professional services functions, to look at our operations and to report on them and to improve their sustainability. Everything from energy managers to people who work on waste, um, people who green disk who work on greening our IT services and so on. So yeah, very comprehensive kind of all of organization approach. And I think that was rather unique about KCS. Typically, environment and sustainability in British universities anyway has been sort of handled as a professional service function. They often have directors of environment who sit within estates and facilities teams. We felt at King's that what we needed to do was really start from the academic mission, have it led by an academic, and then have an all-of-organization approach, which was connected as much as possible. Now, clearly, within that, research plays a very important role. So in Strategy 2026, obviously being refreshed at the moment, we'll have a new Strategy 2030 coming out quite soon towards the end of the year, in which there is a substantial sustainability component as well. But in Strategy 2026, we certainly argued that we wanted to scale up multidisciplinary research to respond to the climate and biodiversity crisis and to embed sustainability into our teaching partnerships and impact, while also leveraging our convening power. So that was the ambition. And we have a number of existing centers of excellence, and of course the Net Zero Center is one of those, but they really exist, increasingly across the university, and they're areas where we think there is potential for greater critical mass, and more connectivity as well across these different centers of excellence. Now, beyond these, of course, there are many researchers who work on these kinds of issues, whether it's in our dental school, in the law faculty, in engineering, who are also doing work. So these centres are not at all the complete picture. So what we try to do within KCS is to create an enabling environment and funding that will allow us to scale up the amount of high quality research which is going on in relation to climate sustainability, as I say, right across the university. We don't want to have this siloed within a sustainability institute. We believe it can happen right across the university. One of the primary instruments we have is this KCS Seed Fund, which is very much modelled on the successful Kings Together model. I'll talk a little bit more about the Seed Fund, how that operates. Obviously, we give researchers developing bids, particularly external bids, support through our RMID. We've run within the research function of KCS a number of workshops to do with health and climate, for instance, various other broad fields where we try to bring people together, networking opportunities, try to stimulate new collaborations and also research, of course. And we're drawing on, recruitment that's going on and the hubs and centres that are there. The new context, I suppose, since last year is also the Concordat for the environmental sustainability of research and innovation, something UKRI worked with other major funders like Wellcome to develop. And King's is a signatory of that. That places some light, but some obligations on us, certainly to report on what we're doing and to continue to incrementally improve the doing of research and innovation, making sure that is also sustainable. So I thought I'd quickly, and I want to thank Adelia for these slides, quickly give a summary of the Seed Fund. We've had 4 rounds. In fact, this morning, before this seminar, we had the panel meeting for round 5. So we made some decisions this morning about spending an additional sum of money. I can't tell you, as you can imagine, anything about what was decided. But Adelia assures me that um, the news of what the outcomes of those were will be with everyone who applied very soon. But as you can see broadly, what this table is trying to show is that actually over time NMES has submitted quite a lot of proposals and actually been pretty successful. So that if you look at round four, um, uh, 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 you know, of the four awards, three involved NMES and attracted about 62% of the total funding. So NMES, I think, has been extremely successful in seeking funds from the seed fund, and this is as we'd expect it. I mean, obviously, there's a growing community of researchers there. You've got the Net Zero Centre. You've got many opportunities, I think, from UKRI and other funders for further follow-on external funding. So it makes sense that NMES has been successful. And then just a few examples of projects that have been funded. So it's broadly in the sort of transport area. In the first round, there was some work on cryogenic fuel liquids in aviation. There's a study that was done on drone delivery fleets and noise and energy, and urban deliveries of carbon bikes. In the sort of broad new materials and new process materials, there's Sarah Barry's project on chemoenzymatic process and drug discovery and development, and Soren and Guy's, and we'll hear from them, project on developing alternative fats using an innovative experimental setup. And then there's a number of projects on energy systems, on semi-transparent solar cells, on solid-state batteries by Laura Lander, and a digital twin for a fusion power plant with Mark Ainsley. So you can see quite a broad set of really interesting projects that have been funded in the previous four rounds, and we'll see what round five brings. And here's another one. Okay, here's some more. Sorry. This is to do with energy systems and net zero. It's a green AI project, carbon trackers, and the project that Raul Rosales has with Westminster and Barbara Sholok and Wei He on heat decarbonisation business models and technology. So, as I say, a really interesting set of projects and hopefully capabilities that are being developed and leading to external funding in the future. So this is kind of who we are. This is what we look like. Actually, I can see at least one person who's no longer with us. She's doing a master's at Oxford now. But this is what we look like in the team. Rachel Mills in the centre as the key sponsor, of course. And this is how you can find us. And I'm going to stop talking and stop sharing there. We're happy to answer any questions at all about KCS. Thank you, Grazia.
Thank you for this very complete overview, for giving us a bit more of context. So as you mentioned, today we have two academics from King's who were awarded one of these King seed funds. So we have Guy Carpenter and Sorin Christian Vladescu. And I just think they're going to talk about their projects about developing alternative fats for healthier, more sustainable food products.
Yeah, great. Thanks, Gracia and Franz. So I'm Guy Carpenter. I'm going to give like an overview of this collaboration, where it's come from and where we're going to. And then Soren is going to present some of the results from the study that's being funded by the seed fund. So how did it? So I'm a professor of oral biology in the dental school, or FODOX as we call it, and I've been studying taste and mouthfeel for some years. And one of those mouthfeel aspects I've been studying is something called astringency, which is the dry mouth sensation you get when you drink red wine or black tea, for example. And it was whilst sort of studying those sort of processes that I got contacted by somebody from PepsiCo, who was Stefan Bayer, and he was interested in forming or funding a study to look at the reduction of sugar in soft drinks for PepsiCo. So he brought together myself and Tom Reddyhoff at Imperial College, and Soren was the postdoc at the time who was working on that project to try and look at how we can replace real sugars or plant-based sugars, let's say, with artificial sweeteners. And so that collaboration continued for a number of years, and it was really interesting because that was the first time that I'd worked with an engineer or a tribologist. Tribology, by the way, is just the study of friction. And so we were trying to sort of understand how it is that when you drink a sugary drink or a carbonated drink containing artificial sweeteners, what sort of happens, what sort of interactions occur in the mouth to then give you that sensation that's, you know, is either pleasant or unpleasant, that sort of drives liking and those kind of things. So that was the sort of background to our collaboration. And since that time, Soren has got an appointment at King's. That's been a great coup, I would say, is to bring some very specialist mechanical engineering skills into King's, and that's formed now into that Department of Engineering. So that's been really good. So we started, as well as looking at sugars and that kind of thing, we've also started to think about how it is that we perceive fats. And around 23, 24, we were sort of in discussions also with Mondelez, which is a company that owns Cadbury's and makes lots of chocolates. And of course, chocolate is a very high fat, high sugar thing that we actually really like eating. So somehow they've the formulation of chocolate sort of defies belief, really, because if you knew that it contained as much sugar as it does or as much fat, you probably wouldn't eat it. And yet it is still one of the most popular snack products out there. So it's quite an achievement to create chocolate, but then it's even more difficult to sort of try and understand how we perceive it, and also how you might substitute things in. So Mondelez are always keen to try and put in, shall we say, more cost-effective ingredients. And so that often leads to reformulation. And I'm sure many of you will remember the Cadbury's cream egg reformulations that didn't go quite so well. So it doesn't take much to, you know, subtly adjust chocolate so that people notice it and don't start liking it. But anyway, it was because of that sort of interest in sort of understanding how we perceive fats that Soren and I applied for this seed funding. And that was to try and look at, how fats in a different product, plant-based milks, is perceived. And that's another really interesting area because, as you know, probably we've been drinking cow's milk for a long time, and people can readily perceive really small changes in fat, whether it's full fat milk, semi or skimmed. You know, the difference there is only 1 to 3% of fat. So humans, we're very, very sensitive to changes in foods. We're very good at perceiving differences. So again, we were really interested. And then also we've had recently a lot of plant-based milks, starting with soya, but also oat, and there's various other plant-based milks. And what they do basically is just to add protein to emulsify the fats that they put into those liquids. And then they might also adjust it with more or less sugar to try and avoid astringency and off taste and those kind of things. So that's why we were sort of interested, is to try and look at how those fats in plant-based milks are perceived in our model of salivary taste, I suppose. So I suppose my USP is that I'm good at purifying proteins or understanding what the proteins in saliva do in the mouth. So that's probably my contribution. And this seed fund was really good because it allowed us to explore a completely new topic area that we didn't have any previous funding for. So it's often a problem, you know, it does happen occasionally that somebody from industry will say, here's some money, go and investigate this problem. But actually the reality most of the time is that you've got to go out there and sort of sell your wares. And for that, you need to have some preliminary data. So to go into a new area, we need to, or to get funding, we need to sort of have some preliminary data. So that's why this seed funding has been excellent for this particular project. And we got some interesting results. And the other good output from this collaboration is our future funding, because during this sort of project, we saw this call for this UKRI cross-disciplinary grant call, and this project is all about how one field can influence another field. So in this case, it's all about how biology, proteins, salivary proteins, influences engineering and vice versa. So this is a really quite high profile grant call, and the first round was very popular, and it had something crazy like 1,000 applications or something. And so they had several rounds of thinning out, and amazingly we got through both the King's thinning out process, where they only selected, I think, 20 or so applications to go forward. And then also the UKRI also did a thinning out where they got rid of 80% of the applicants. And so we're into that final phase now where we're just about to submit in a couple of weeks' time. So this is for 2 postdocs for 2 years. So it's quite a short project, about £1 million probably. But this is to sort of look at some of the spin outs coming from that seed funding and from other things. And so one part is it to look at how we can replace fats in foods, and that's again using some of the data from the seed funding. But the other one, which I'm really interested in, is actually looking at how mucins, so those are the sticky substances in saliva, lubricate, because what we've shown is that mucins are very good at trapping ions, so sodium ions specifically. And because they can do that, they form this very lubricating hydration layer, we call it. And we want to try and exploit that into making a new class of lubricants. So most oils are petrochemical derived, and so the idea here is that we can move away from petrochemical lubricants by exploiting this what's called hydration lubrication, which is something that proteins do. And of course, some of the early lubricants were plant-based. So you think of castor oils, for example, they have a lot of proteins from the seed husk that help to create these oils. So plant-based, not plant-based, aqueous-based lubricants have a potential application, particularly for things like electric motors in cars, electric cars, because that lubricant also gets used to circulate around the electric motor and helps to cool the motor. So this sort of new class of aqueous lubricants is very new. And so we think we've got an interesting insight or niche that may be able to be exploited. And so that's why this cross-disciplinary maybe, hopefully, will be successful. But like I say, it also uses some of the data from the seed funding that we had. So maybe I can hand over to Soren to show us some of the results from that.
Yes, thank you very much. I will share my screen now. Okay, yeah, thank you very much. So I am Sorin Vladescu. I'm from the Department of Engineering. I'm a lecturer. And yeah, as Guy mentioned, we got this seed fund in round 2. All the work was done by our colleague Kyang-Kun Yap, which is an amazing researcher we hired for 9 months on this project. And I'm going to show you some of his results on this project. Before starting, I just want to mention that with the results from this KCS fund, Yap went to an international conference for food oral processing in Valencia last month, and he got the best poster presentation award. It was just one prize and maybe 80 presentations. So yeah, this is from the newsletter we had in the department. Regarding the problem, I will very briefly discuss, because Guy also mentioned the main problem related to climate and sustainability is the fact that over the next 30 years, there is a projected increase of 2 billion people. And of course, there is going to be a significant demand for animal proteins, which will accelerate carbon emissions. And according to BCG study, which you can see down here, plant-based proteins are the best way to reduce these climate emissions. For every dollar invested, you can achieve 11 times more reduction than, for example, electric vehicles. However, there is a problem. There is a significant difference both in fat and lean between animal meat and plant-based meat. And as Guy mentioned, there is a significant limitation, the fact that the mouthfeel and the perceived astringency limits the adoption of these products. So our main goal is to understand how both animal proteins and dairy proteins and animal and plant proteins behave in the oral contact. And what we are trying to do, as I said, we are trying to simulate the oral contact using a tribometer, which I will show immediately. And our unique approach, let's say, is the fact that we also look inside the contact to see exactly what happens, how these animal proteins or plant-based proteins act to remove the saliva so that they can reach the taste buds. And yeah, then we are trying to analyze how this variation in oral lubrication and oral friction can be associated with sensory characteristics, as you can see in this image from a different study, the one that Guy mentioned at the beginning. Yeah, so the way we want to do it, or the way we did it until now, was using this setup A, which you can see here. It is a custom tribometer. However, we modified it in such a way so we can look inside the contact. For in the future, we want to build our own, which will have a complex and dynamic movement and also build a biomimetic tank. However, yeah, this is for a future grant application. Today, I'm gonna show you our results obtained with this tribometer, which, as I said, by using this microscope, allows us to visualize the contact, see how different saliva proteins behave, how plant proteins behave, and yeah. So I'm not gonna spend too much time on this because I'm gonna show you step by step how we do that. And I'm gonna do this by using Yap's presentation, the one I mentioned at the beginning, where we are gonna use tribology and in situ fluorescent microscopy to characterize plant-based milks. Animal milk has long been a major source of proteins, as we know. However, there is a growing interest, as Guy also mentioned, in plant-based milks. And this is due to different ethical or health or environmental reasons. And to fast-track consumer acceptance of these plant milks, we need to better understand and improve their taste and texture. And one way to do that, as we mentioned, is through tribology, which is the study of friction and lubrication. In this case, the friction and lubrication inside the mouth, so the oral friction and lubrication. And yeah, these parameters are often related with sensory perception. And how we run our experiments, we have a pin on disc setup to study the friction of these milks and their behavior inside the contact. So we have a pin, we have a rotating disc. Both of them are made from a special material, from PDMS, with a certain roughness and a certain elasticity, so it can accurately mimic the surfaces inside the mouth. And then we, during each test, we add different milks. The pin is connected to a sensor, and what's special, as I said, about our setup is that we have a fluorescent microscope under this transparent disc, and we can look at the contact, see what's happening in the contact in real time, and then link these images, this visual data with actual friction data. And we do that for both cow and plant milks. For cow milks, we've selected skim, semi-skim, and whole milks. And for plant milks, we selected the most popular ones, soya, oat, and almond. And we run different set of experiments. I'm going to go very fast with the experiments, but just, yeah, we run one set only with milks to actually understand the friction of milks, and one set of experiments where we first create a layer of saliva on the surface by rubbing this saliva for 10 minutes. Then we remove part of it. On top of that adhered layer of saliva, we add the milks, and we measure or we record the delubrication process, both visually and in terms of friction. Yeah, in this graph, we have the friction results for all the milks we've tested, both with saliva and without saliva. And yeah, gray bars are the results only for milks, orange for milk plus saliva. Of course, there is a lot going on in this graph, but I'm just gonna focus on the main findings. And basically the significant difference between just milk and milk plus saliva, especially for the skim milk and for the oat milk, where we can see a significant decrease in friction for the skim milk when we have saliva, and a significant increase in friction for oat milk, again, when we have saliva. And this highlights the importance of always running our experiments using real human saliva, for, yeah, which we have all the ethics in place. Now I'll focus only on the results with the saliva because they are definitely the most relevant. And we want to see why these skim milks and soya milk have such a low coefficient of friction, oral friction. And in order to understand why this happens, as I said, we have this optical setup where we can look at the contact to understand what's happening. And how we do it is by dye tagging the milks with different dyes. And before discussing about the dyes, this is the contact. So with the dotted line, we have the contact, which you can see here, and we have the inlet. So basically the way the milks flow inside the mimic oral contact. And as I said, we use two different types of dye. One FITC dye, which tags hydrophilic substances, so more watery parts, and one dye called Nile red, which tags lipophilic substances, so more fatty parts inside the milk. Now, for the case with the FITC dye tagging, we found that we have these substances in all the milks, so no real correlation between the friction results and the optical results. However, for the lipophilic dye, the Nile red, we can see that the friction is high. If you remember the graph with the orange bars, when we have these lipophilic substances, and the friction is low for the skim and soya milk. So when these lipophilic substances are missing. And yeah, we definitely have a correlation between this friction results that we get, the oral friction, and whatever we see that is accumulating, let's say, inside the contact. And yeah, we believe that these substances are basically blocking the contact, blocking the inlet, which disrupts the lubricating salivary film or the milk film, which leads to an increase in friction in all other cases except skim milk and soy. Yeah, so these are part of our results. As Guy mentioned, using these initial studies and the setup that we developed for these studies, we were able to apply for the cross-disciplinary grant that Guy mentioned. We got some, Guy got some funding from Mondelez for 3 or 4 months study on chocolate or fat for chocolate. And we also applied for a Good Food Institute grant to develop that new tribometer with a complex movement, which we just submitted last month. And yeah, thank you.
Okay, thank you very much to the speakers thank you for this very interesting project. I think this is really quite different from the typical webinar we have here, and it's a great example of interdisciplinary research. So thank you very much for sharing that. So now we move formally to the Q&A part of the webinar. So I think feel free, yes, to raise your hand and or to write your question in the chat. And then I will try to go to moderate in order. So I think we have a question now from Peter Dobson.
Thank you. I was really interested in that because I myself am trying to convert a tribometer at the moment with different microscopes on it. So I was really interested in this. And my first question would be, you're using saliva, but saliva contains enzymes which themselves are proteins. So have you quantified the enzyme content of the saliva each time you do the experiment? I think that could be really important. I have many other points to ask about microscopes, but I'll limit myself to that question.
Sure. Well, maybe I can answer that one as a salivary Waller. Yeah, that's right. There's lots of proteolytic and glycolytic enzymes in saliva, mostly coming from the bacterial or the microbiome of the mouth, which is significant. But also some of them are mammalian based, so our own enzymes. And so saliva is highly dynamic. So it changes almost as soon as you collect it, like within minutes, we can measure certain physical properties are changing. But we have characterized that. And then we've also got around that by purifying out the proteins of interest, which are the mucins. And so analyzing those in the absence of proteolytic activity. So yeah, it is a highly dynamic substance, and I think that's why a lot of people avoid it. So they don't study it because it is complicated. But yeah, we certainly looked at those proteolytic and glycolytic enzymes. Yeah.
Thank you. I'm also involved in a project which is trying to analyze biomarkers in saliva, and the big issue with that is the enzymes and the proteins that are already present. They can completely modify the biomarker you're looking for. So I wonder, have you got a protocol to develop what I'd call a standard saliva sample?
Yeah, there are protocols and ways around those proteolytic activities, and the obvious one is to collect it quickly and put it on ice. That you can add inhibitors, of course, of proteases and that kind of thing. But yeah, there are protocols. Yeah, Peter, if you contact me by email something, we can definitely chat about that. That's no problem. Yeah.
Yeah, I would like to, and I'll tell you a bit about the work I'm doing on the microscope side as well.
Sure, that sounds good. Thank you.
Yeah, Franz.
So this isn't my field at all, so I'm going to ask probably a very idiotic question. But there seemed in the data you presented, Soren, to be a marked difference between animal milks and plant-based milks in the sort of the friction that's. I'm presuming that's a well-known result, but what explains it and is perhaps one other audience for the work you're doing. The plant-based milk producers who presumably want to mimic the mouthfeel of cow's milk or goat's milk or whatever. Yeah.
Mm-hmm.
So I don't think I understand the question. Sorry, Franz.
So the question, I suppose, is the results you've got suggest that there really is, I mean, a measurable difference.
Yeah.
In the friction and therefore the mouthfeel of these different classes of milk.
Mm-hmm.
Are there things you can do with the plant-based milk to change its characteristics so that it's the way in which it operates in the mouth, is more similar to animal milks?
Yeah. So in this first part of the study, we kind of try to understand how these different types of milk behave, because there was definitely no study to measure this before and actually to look and understand what happens. So they are not really common results, let's say.
Okay.
We had absolutely no idea how they behave, and now we are trying to understand why they behave in such a way. For example, for some of them, friction increases when you add saliva, for some of them decreases. And yeah, we are more trying to understand what is there. For example, we tried initially with that FITC dye, however, it was tagging everything and you couldn't actually understand any difference between them. And that's why we started to try to tag only the fat part to see any changes and any correlation. And yeah, it's kind of ongoing.
And also, just to add to that, I think it's about how the proteins emulsify the fats. So different emulsifiers are used. So in chocolate, for example, it's called lecithin, which is a funny name for chocolate. And but in these plant-based milks, there is the proteins coming from the oat or the almond that are helping to emulsify the fat, so that they. But of course, what the makers of these things are doing, they're formulating their product so the product is stable. And then they go through a sort of empirical process to see if people like it. So what we're trying to do is to go back the other way. So we're trying to take those ones that are market leaders and then back engineer it to work out why it's successful. And the way it seems to be looking is that successful ones are the ones that emulsify fats the best. So that if you have a poorly emulsified fat, then you seem to get a fat spreading in the mouth, and that leads to that sort of oily texture that some of these plant-based milks have. Some people like that, of course, so that's another problem, but it seems to be down to the quality of the emulsifier, and saliva itself is also an excellent emulsifier. So these mucin things, they seem to interact with these fat droplets as well, and so it's complicated.
But it's what you're suggesting. Sorry, just to follow on a little bit. Is it really is that crude that Cadbury's or Mondelez or whoever, they just feed it to people and they collect reactions. They don't actually have any biological or chemical explanation for. Okay, so this work is really pretty cutting edge and novel. Yeah,
that's right. Yeah, so plenty of product formulation knowledge, but very little human physiology type knowledge, I would say. Okay. And so I think that's our niche in this project. Yeah.
Okay.
And just to add, maybe these companies focus a lot on sensory panels. However, those sensory panels can be a bit random sometimes, or not extremely precise or repeatable. So with this technique, we are kind of trying to get to rank all these products in a more organized way.
Thank you. So maybe now I just want to ask maybe a bit more of a general question, just more about maybe your collaboration in general, right? Because obviously here we have collaboration between the Department of Engineering and the Faculty of Dentistry, Oral and Craniofacial Science. So they're very like distant field in a sense. And I just wonder if you maybe can make comments or suggestions on maybe challenges, because we see this is a success story. You have very good results, applications, it works well. But I think it's also maybe interesting to know maybe some of the challenges, maybe if you can give tips to other researchers that maybe want to establish this type of collaborations.
Guy, maybe you can discuss about you and Tom, because I already.
Yeah, it's interesting, isn't it? Collaboration. It seems quite a hard thing to achieve, and then once you achieve it, you sort of think, well, what was all the trouble about it? I think it seems to be that each field has its own language, and so trying to talk to somebody else about that language, or rather without using that language, is a good first step. So, but quite often, when you, like in my field, saliva, nobody wants to investigate saliva. It's a fairly disgusting kind of topic. Nobody particularly wants to get, and yet it's got loads of really interesting proteins and functions and those kind of things. So so nobody wants to talk about saliva, but everyone loves talking about food. So as soon as I translate it to food, everyone's like, oh yeah, I get that. And so we would doing stuff about carbonation of drinks and how you perceive that, and people's all interested in that. And then artificial sweeteners, we're all interested in that. And then so from my point of view, I just have to explain it in simplistic terms, I suppose. And so not simplistic terms, that's just changing the language, I suppose. Common language.
In my case, I think I'm just lucky that this tribology, so the friction and lubrication between two surfaces, can be applied in many, many different fields. I know, from hip joints replacement to internal combustion engines to food, oral, and
Yeah, that's also very interesting. Yes. Thank you. Very good insights. There is a question from Delia. I think for both of you, have you considered working with colleagues in the nutritional sciences?
Yeah, of course. They don't tend to be so interested in the oral perception side, strangely. So I haven't had that much enthusiasm from them, but they're sort of more interested in the digestion part. So they're sort of get it past there, and then we can talk. So yes, we certainly explored some initial interactions with it and sort of similar sort of ideas, I suppose.
Yeah, thank you. And maybe I just want to ask a question to Franz. Sorry, I see that you type in a question.
Yeah, I was interested in the work on mucins. I'd never heard of mucins, so I had to Google that one. But I love that idea that these could, so if we could understand how they lubricate, they could lead to a whole new class of lubricants. I mean, who knew that, you know, that the answer to many of our questions is hiding in our mouth, So is that a serendipitous? I suppose my question is that a serendipitous outcome of the work you've done with Sorin, because you've got this tribology going on, or did you know that already and have always suspected it?
No, I've been working on mucins mucins for a long time, and as one of the salivary proteins. So there's lots of different types of salivary proteins. But yeah, it's only because I was working with Soren and Tom that we got this strange result whereby if we completely dialyzed away all the ions, so with proteins, quite often you have to have ions there so that they remain soluble. But if you did dialyze them away, then it had very high friction. And then if you put the ions back in, friction goes right down. And now we're getting to like really low levels of friction, like they call it super lubricity, but I wouldn't have known that obviously without working with Sorin. But yeah, so my background is looking at dry mouth. So again, you'd think that that's because there's not enough saliva. That's true. There's also this group of people that have plenty of saliva that doesn't work very well, and so it's quite a common complaint, xerostomia. And so but yeah, it's sort of interesting now because just that sort of basic interaction of how ions interact with proteins is quite fundamental to like loads of different things. And so now we're studying in loads more detail. And it turns out there's lots of other people who are interested in that sort of field, but for completely different reasons. So it makes your work a bit more relevant and mainstream, as opposed to sort of peripheral stuff. So yeah.
Yeah, and I think it's also very well timed considering the expansion of these electric vehicles, because with these electric vehicles, you use the same fluid to cool down the battery and to lubricate the gearbox and whatever bearings you have for the electric motor. You can't really cool down with oil, with a petroleum oil, so you need a more less viscous water-based type of lubricant. However, with good enough properties to also protect your gears, let's say, while cooling down the battery. So I think maybe this is something useful and very well timed.
Great, thank you. Maybe just have a quick question for Franz before we close, because I think hopefully now more people know about the seed fund, and I think you have another round, I think is on your table coming up this year. And I just wonder, maybe, I know you cannot say too much, but maybe if you have some tips of what makes a successful application to this seed fund, and maybe what should be avoided.
Yeah, so we do have another round, and in fact, it'll open quite soon in July, I think. The deadline will be in October. And that'll probably be the final round in this batch. But as I say, we're talking about extending KCS, and as part of that, we very much hope there'll continue to be a seed fund, and therefore we expect to be able to support this kind of exploratory work into the future as well. What makes a good seed fund? I mean, we've supported quite a variety of different things. I think what people are looking for generally is a really interesting idea, a really good, often multidisciplinary group of people looking at it. So Soren and Guy, are a sort of exemplar for that, and a clear pathway to funding. And the project shouldn't be too big, you know. So I think sometimes projects fail because they may be really interesting, but the proposal that has been written for, I don't know, £60,000 or something, feels like a massive amount of work that probably couldn't really be done. So people they're packing too much in. So keep should be a pilot project, developing some initial data that will enable you to write the bid for the external funder for the big thing. So good idea, keep it kind of simple and direct and concrete, a good partnership, and a clear pathway to funding that is credible. I think that's what it should be. But you know, it's a very broad panel, and you know, we funded lots of different kinds of things, and so we do emphasize diversity as well. And yeah, if you have a good idea, whatever it is, please come forward and do work with the team, work with Adelia, work with Jacqueline, work with other you know, other you know, research and development managers, to develop the idea. I have to say the proposals we saw this morning, in general, very, very high level. So there's really good stuff coming through the pipeline.
Okay, great. Thank you. Thank you for these insights. So I think now people are tuned to the next call. So with this, I formally close the webinar. I thank all the speakers for their time and all the attendees. And I just want to mention the next webinar, the last one of this academic year, will be on July 9th. And thank you again, and have a good rest of the day.
Thank you very much.
Thank you.
Bye-bye. Yeah. Bye-bye.
Thank you.